From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 21, recorded on August 16th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from San Francisco, Ori Lieberman.
Hi, Vincent. How are you doing?
Hey, welcome back. We missed you one episode, I think.
Yes, thank you. Well, one episode and then one episode with bad sound, and I got logged off halfway through, so
yeah.
Making my triumphant return.
And you're enjoying San Francisco?
Yeah, mostly in the hospital, so seeing a lot of the city, you know.
Great.
Yeah.
Also joining us from New York, Tim Chung. Hello, Tim.
Hello. Hi, Vincent. Hello, Ori. I hope the mic is okay.
Everything's good. Everything's good. And this is TWiN. We're going to talk about your brain. First, Ori has an announcement to make.
So we mentioned this a couple episodes ago, but we'll post the link to a listener poll on the show notes just to get a little bit more information about who's listening, how we can steer our conversations to best suit the audience. And there will be a prize at the end that we will, to be decided prize if you put your email address in to incentivize people to fill it out.
Oh, that could be interesting. A prize.
I'm sure what
a known prize.
A piece of a brain. You never know.
It could be a nice pen with a TWiN logo, right?
Yeah, that would be great.
Yeah, that would be a cool prize. I'd buy one of those. Nice. All right. Today is Tim's turn to educate us. Take it away, Tim.
Well, we'll see about the educating thing. But all right. So today, the paper, we have an interesting paper. And it's the paper promises us that it can tell us whether a mouse is hallucinating, which obviously is interesting. Well,
that's a good question, Tim, because I can imagine saying, who cares if a mouse hallucinates, right?
Whether you can even tell if a mouse is hallucinating by itself is fascinating.
Mm-hmm.
But of course, there are people who suffer from hallucination, and that would be the most relevant reason to understand the origin of hallucination. But to kick us off, I'd like to ask Ori a question since Ori has returned, and I can pick on him momentarily for this week. So Ori, welcome back.
Thank you.
Now, I would like to ask you a question. How do you know right now that you're, instead of recording TWiN, you're not just dreaming that you're recording TWiN, and instead of now being middle of August and you're on holiday, it's actually early July and it's now 4 a.m. and you're missing your early grand round in the hospital. And you really should be waking up because you're late.
Well, I'll tell you one thing is that whenever I have bad dreams about that, I usually don't have my pants on, and I have pants on. So we know that it's reality.
That is a good check. But it might, let's say you're not able to see your legs, then you can picture it being quite hard to tell whether you're dreaming, or you can try to pinch yourself awake. But to whether to tell you yourself are hallucinating can be quite difficult. If you think about it, it might be easier to tell whether your friend is hallucinating than whether you yourself is hallucinating. Because if your friend is hallucinating, they might claim, no, I actually did hear a voice, whereas you would say, no, you didn't. And all you have to do is to ask another friend whether they also heard something. And if several of you agree that there's no voice, but your friend who might be hallucinating claim there's a voice, and claim it with confidence, then you might start to wonder maybe your friend actually is hallucinating. So based on that, you might expect it's easier to tell whether someone else is hallucinating than whether you yourself are hallucinating. But what might then be harder is, can you tell if a mouse is hallucinating when the mouse can't really talk to you? So this is why this paper is very interesting, because this paper seems to have come up with a strategy to test whether a mouse is hallucinating. And as Vincent alluded to, why do we care about whether a mouse is hearing voices inside its head? That's because one of the symptoms, one of the indication of hallucination is actually schizophrenia, or schizophrenia in the US. And one of the symptoms is auditory and also sometimes visual hallucination. And you hear voices inside your head to compel you to do things. And well, Ori, you are an actual doctor, so please correct me if anything I say is incorrect. I'm the wrong kind of doctor, so I don't know what I'm saying, really.
Tim, can I ask you,
sure.
Can you define hallucination? Just put us all on the same line.
Hallucination is when a patient comes in. So I cannot define it very well. So I don't have like the diagnostic DSM-5, the diagnostic criterion manual in front of me. But my guess as a non-doctor would be if a patient comes in and claims to be hearing voices when you of sound mind think there's no voices and other people agree there's no voices, or other things. It could be visual hallucinations, so like seeing patterns and stuff, or faces, where the actual other people decide that is not real, then that would be defined as hallucination, I think. I don't know whether I assume that is, it will be something along those lines.
And can I just add one thing to that? Sure. So you said that someone comes into hospital with hearing or with a sensory experience that's not based in reality. But I think what this paper highlights in the middle during the human part of the study is that hallucinations are happening on a regular basis to a wide spectrum of people and are not necessarily associated with clinically significant psychiatric disease. So, and I think that one thing to remember is that, now this is my West Coast in me talking, but we talk about normal and abnormal, but these are all on a spectrum. And what we think about is impairment. So are your daily activities impaired from hallucinations that could lead you to go to hospital? But in this study in particular, we are actually looking at hallucinations in the general population, less so in an impaired population. And I think that will be something that we can talk about later on after we go through the details of the paper, but are important to remember.
Is a dream a kind of a hallucination?
I don't know. Daydreaming, I don't think is hallucination.
No, but during sleep, I have dreams. Sometimes I remember, and they don't make any sense, right? Things are not normal. I step out of a plane and I'm fine, and things like that. So it's I can't tell if I've ever hallucinated or not. So I would guess that's the closest that I've come. I don't know if that's correct or not. I'm just, Ori, this is your purview, right?
Well, my purview is or my future potential purview is in when hallucinations impair people's function. But
yeah,
I guess I would say that, there are moments in my life where I wonder whether I've hallucinated, when you hear a cat going through the room when you have cats, but the cats are actually in the other room, or you hear someone you think called your name, but there was no one actually there. And, these are sensory experiences that you feel internally, subjectively, but are not objectively true. And they have very characteristic properties. So, and I'll illustrate that by going back to the cat example. There was a period of time when I had moved the cats out here, but had not fully moved out of my apartment, and returned to New York to move out of the apartment. And it was the first time that I had been in this apartment without the cats. And my God, I thought the cats were everywhere. I would turn around, I would hear a noise, or I would see a movement in the corner of my vision. And because my brain had been trained to expect these things in this environment, and the cats were not there, right? So hallucinations can increase in frequency if you have a very high expectation that sensory experience is actually true, even if it's not. Not to mention the emotional aspects of moving out of an apartment in a city, which I had called home for 8 years, that perhaps made me long for a time when the cats were there. And, you can get into all these psychological and spiritual interpretations that led one to have sensory experiences that might not have been true.
Yeah. But to address whether dream and hallucination are similar, I think the answer would have to lean on some sort of neuroscience studies to look at whether the brain looks similar under the two different circumstances. And I don't think we know enough yet.
Okay.
Anyway, so some background about what the state of schizophrenia researchers prior to this paper, which is in preclinical stuff. So we like to have animal models of neuropsychiatric disease because we can more easily experiment on animals to find out the root cause, to find out what change in the brain kind of correlates or also actually causes different kind of psychiatric illnesses. And for psychiatry, this is particularly difficult because there's no good animal model of, say, hallucination or psychotic break. And for instance, one of the very well used model is this model called pre-pulse inhibition for schizophrenia. And in that model, researchers found that in most people, if you give them a really, really loud noise, they would freeze. They would have this startle effect. They would what just happened? And their hairs would stand on end and stuff like that. And the same for mice. They also have this startle response. But if you were to play a very, very, a much milder tone right before the loud tone, then in most humans, the startle response gets suppressed because your brain kind of adjusts. Your brain kind of gets prepared that something else is coming. But this startle response suppression, also known as pre-pulse inhibition, is not seen or is reduced in schizophrenic patients. And if you give them antipsychotic medication, it comes back. So this is one of the model for people that people have used for like 20, 30 years, maybe 40 years for schizophrenia. But if I'm going to say no patients turn up to the doctor and say, "Doctor, I have a problem. My pre-pulse inhibition response is gone." Whenever there's a loud tone that was preceded by a small tone, I still startle. That doesn't happen. So from that, you can tell that the schizophrenia model doesn't really have much of what we call face validity. It doesn't seem to model the symptoms very well. And ideally, what we'd like is to be able to have an animal model of hallucination so that we can play around and look at what happens to the brain when the mouse is hallucinating. Sorry, I have a question.
I guess maybe we could just formalize the different types of validity that animal models could have.
Do you know?
So I can do that. And so I think about animal models having 3 different types of validity. So number 1, which you have been talking about, is face validity. So that, for example, you would say a mouse is hallucinating. It could tell you that it's hallucinating, and therefore it's a model of hallucinations. Okay. You can also have construct validity. So in construct validity, you are modeling some form of what is thought to be an underlying mechanism in another animal, which you can then experiment on. So an example of this is putting a genetic mutation that we see in a disease state in humans into a mouse, and whatever phenotype manifests in the mouse is an animal model of this disease, and that mouse model has construct validity. Mm-hmm. Okay. The last one is predictive validity, and this has really driven the vast majority of neuropsychiatric research for drug discovery over the last 60 years, let's say. And in this model, essentially, we take drugs or molecules that have efficacy in humans, and we see what behaviors are perturbed in an animal, these could have no relationship at all to the behaviors that are perturbed in humans. And then we can use that and say that any new molecules that affect that behavior in animals we would predict would have efficacy in human behaviors. And for example, a lot of antidepressants were shown to work on a forced swim test. So this is a nice example of predictive validity. So in this test, a mouse is thrown into a water pool, and eventually they will give up and stop trying to swim and get out. And this is what's called learned helplessness. And many antidepressants increase the time that a mouse fights to get out of the water. And these then have efficacy in treating depressive symptoms in humans. But this has no face or necessarily construct validity with major depressive disorder in humans. But we use it to screen for drugs. So pre-pulse inhibition is actually a model that has predictive validity in schizophrenia, because antipsychotics increase pre-pulse inhibition in animal models, or in animals. Interestingly, pre-pulse inhibition also has construct validity in some situations. And the way I think about that is that there's actually a really nice paper in the early 2010s in Science that showed that D2 receptors on an auditory cortex circuit mediate a change in pre-pulse inhibition after pretreatment with amphetamine, which we know can cause psychosis in humans. And correcting that by giving antipsychotics, which target these two receptors, normalize the circuit function. So this model has some predictive, maybe some construct validity, but obviously no face validity, as you said. So in this case, we're talking about actually developing a model that has some face validity for hallucinations, right?
Mm-hmm, mm-hmm. And then starting from there, and hopefully the predictive and perhaps construct validity would follow, would be my guess. Because I'm guessing that ideally you have all the validities. That would be a perfect model.
Unfortunately, as we've talked about in the past on the show, we did a TWiN this winter about Alzheimer's disease. And these models have great construct validity because we can model genetic mutations that we see in Alzheimer's disease. And they have some face validity because the mice show dementia or memory impairments. But whether they have predictive validity is a separate question, because a lot of the drugs that impact the disease process we see in those models don't necessarily have an impact in humans. So as you can see, that it's not always easy to say, oh, face validity, construct validity, therefore predictive validity. And in the end, what do we care about except predictive validity?
Yeah, but I have a feeling, for example, going back to, so we're kind of digressing a little bit, but it's fun to talk about the things like that, because I didn't even know about construct validity until you told me about this. But for example, for antidepressant and predictive validity, antidepressant works well, and it definitely makes the mouse swim for longer, but it's also known in human that it doesn't work all the time. So even the predictive validity might not be 100%. And it's possible that if you have a model that has more of the other validity, then you would get even better predictive validity.
Of course, these are all balances. So the question is, like, when you have a new model, what types of validity does your model have?
Right.
Yeah. And how can you test that? And I think that this paper did a really nice job of working through these different types of things, and we can go through it as we go through the paper.
Okay, so let's do just that. So let's dig into the paper. So the paper I'm going to talk about today is published in Science. And it's titled Striatal Dopamine Mediates Hallucination-like Perception in Mice. And it is by first author Katarina Schmack, and also by Bosk, Ott, Sturgill, and Kapech. It's from the Kapech lab, who I think are in Cold Spring Harbor, and also WashU St. Louis, I believe. I hope I'm right. Yes. Okay, so this paper comes at the hallucination problem from a completely different angle. It basically comes from the proposal that when a human patient is hallucinating, what it reports is an auditory sound, a sound when there's actually no sound, and that it is they're very highly confident that there was a sound. So these two things together, if you can replicate that in a mouse, might give you a model of hallucination. So how do they do that? They designed this really wonderful experiment, wonderful paradigm with mice. So it's kind of like on TWiN, on This Week in Virology, the hosts like to talk about taking the mouse to the opera to test whether the mouse is sick. And these authors really did take the mice to the opera. And so this is the design of the experiment. The mouse is put in a box, and at the front of the box, there are three holes, three nose poke holes, and the mouse can put the little snout in one of the holes, and there's an infrared detector to detect when the mouse's snout is in the hole. And the mouse is trained to initiate a trial. So the session is divided into different trials. And the mouse initiates a trial by poking its nose into the middle hole. And once it does that, the computer registers it, and it blinks on a light. And half the time, when the light comes on, there's a tone. Oh, I forgot to mention, very important, throughout the entire session, there is a white noise in the background. So the cage is not completely silent. There's like a somewhat loud white noise kind of sound. It sounds like if you're at the bottom of a waterfall, that's the kind of sound white noise sounds like. So the mouse poke its nose into the middle hole, and a light would come on, and half the time there would be a tone that would be played by the computer on top of this white noise background. And the other half of the time, there is no tone. And the mouse's job is to tell whether there's a tone or no tone. And if it heard a tone, if there was a tone and the mouse goes to, for example, one of the port, let's say the right port, and then sticks its nose in, it will get a water reward. So these mice are thirsty and they want water. So if it detects a tone and goes to one of the ports, so for the sake of argument, let's say the right port, then it would get a water reward. If it goes to the left port, there will be no reward. And conversely, if it heard no tone, then it would go to the left port for a reward. And the right port, if it goes to the right port, there will be no reward. But there's a trick that there are two tricks that these researchers do to this seemingly innocuous task. The first trick is that the tone has varying loudness. So on different trials, the tone would be very, very loud, can be very, very loud, in which case it will be a very easy trial. And the mouse can be able to tell that a tone did occur. Whereas on other trials, the tone would be much quieter, and it would be much more difficult for the mouse to hear that there was actually a tone. And sometimes it might get it wrong. So that's trick number one. And trick number two is that once the mouse has poked its nose into the correct port, the water doesn't get given right away. Instead, the mouse has to wait for an unpredictable amount of time until the water comes. And what is even more devious for the researchers is actually a wonderful design, is that on 5% of the trials, even when the mouse is correct, they would actually not give the mouse water. So the trial would go on for as long as the mouse are willing to stay in the port. So the mouse has to invest. It has to, once it's made a decision from the center port, whether it heard a tone or not, and then selected a port, it has to invest an amount of time until the water comes. If the water comes, then it can lick and it gets a reward and it's happy. But if the water doesn't come, it has to decide how long to invest for. And the researcher suggests that the longer the mouse is willing to invest, the higher the confidence. So this would allow them to tell whether the mouse is hallucinating, because if a mouse decides to go to the port that says there's been a tone when there's been no tone. In reality, there's been no tone. If the mouse says there's been a tone and I'm going to wait forever for it, that would suggest that the mouse may have really heard something. So this is all well and good, but how are there evidence? Is there any evidence that the mouse is actually behaving the way that this confidence really does suggest? And from
let me just relate this back to something that we talked about earlier, which is my sensory experience of hearing potential cats in my apartment, right? So these were clear sensory experiences. I heard something, okay? But I had very low confidence that they were real because I knew that the cats were not there, okay? So one could say that these were then not hallucinations in the way that they're defining what hallucinations were. However, if I heard something very loud in my apartment, I would have more confidence that this was actually a real sound. And that is essentially this kind of the trick that they're playing on, that they're trying to see how soft they can make the sound before the mouse thinks that maybe it's real and maybe it's not real, and how confident they are. And this kind of sensory experience plus confidence is what defines the hallucination. And if you talk to someone who has had hallucinations, we had a patient last week who was convinced that Jay-Z was talking to them and ruining their life, and this was real to them. This was a true experience in their mind, even though we all assumed, I guess, that Jay-Z was not actually communicating with them. And I think that is kind of the key in the definition of hallucinations and really the exciting thing that they add in this behavioral paradigm.
Yep, completely agree.
So Tim, this setup works in that even with all this uncertainty, the mouse will invest time and go for the sweet water.
Exactly. So the way that the mouse's behavior suggests that it makes sense to us as human, that the mouse's confidence relates to that it actually heard something, is that when you play a very, very loud tone, what the researchers found is that the mouse is willing to wait for a very, very long time at the port. And when you play a very, very loud tone and the mouse makes a mistake and go to the wrong port, they actually very quickly quit. They kind of realize, oh, what did I just do? I have very low confidence. And as the tone gets quieter and quieter, what you see is that the mouse becomes more and more uncertain.
I see.
That they actually, when they make a right choice, they are kind of like, less sure about this. So that's why the researcher think maybe this kind of, as already said, there's some face validity. It kind of appealed to our intuition about what our perception in our head is reflecting. But what's interesting is that so when the tone is very loud, and if you were to plot, we're talking about figure 1 here, in case anyone has the paper in front of them. If you were to plot how often the mouse reported there's a sound, so like how often they go to I heard a sound nose port. When the tone is very loud, the mouse is almost 100% going to the loud port, the tone port. And as you turn the volume lower and lower to make it harder and harder, the mouse now kind of the choice is creeping down towards more 50-50. And even on trials where there is completely no tone whatsoever, it's just a background noise, the mice are still reporting that there was a tone about 16% of the time. And for a substantial amount of those 16%, what the researchers called false alarm, because there was no tone, but the mouse said there was a tone. So it kind of rang the alarm bell. For a substantial amount of those trials, the mouse stayed there for quite a long time. So suggesting that, as already said, because the experiment is designed such that a lot of quiet tones are still rewarded with water. And some of those quiet tone might have been perceived as essentially no tone at all. the mouse kind of in its head are expecting maybe some of these trials that sound like maybe it's no tone, maybe there's a tone. I'm going to think there's a tone. I'm going to go to the note support.
Is this experimental setup actually inducing the mice to hallucinate?
No, remember, this is the conversation that I wanted to bring up later on in our recording, which is that these are in quote unquote normal mice, right? So they haven't changed the threshold to hallucinate. And this is why I think it's important to remember that hallucinations or our disturbances are kind of present throughout the population at a certain rate, but the rate of them is higher and the confidence in them is higher in people who have primary psychotic disorders. The other thing that I would point out, which I thought was a really elegant internal kind of analysis, was that of the trials where there was no tone, that they poke their head in the, yes, there was a tone port, the time that they wait or the amount of confidence they have actually overlaps with the amount of confidence they have in tones that were very, very soft. So this suggests that they're not just making the wrong choice, right? They're actually confident, and they're just as confident as very, very soft tones. So that suggests that there is like essentially a sensory disturbance that is there, and they have as much confidence in it as a true sensory experience that we know is there because we've actually given them the sound. And then let me just add one more thing, which is that these are insane mouse experiments. So there's one line in and insane also used liberally there, but there is one line in the paper where essentially these mice are trained for weeks to do this task well. Okay. So if you've ever seen a mouse, you can imagine how long and how difficult this task can be for them. So there is an aspect of probably training and maybe some overtraining. And that was one thing I wanted to ask you about, Tim, which was that if there are some kind of, how do you incorporate the non-rewarded trials into the motivation for continued confidence in low sound trials? So for example, if I think I hear a low tone, okay, and then I stick my head in the port that says there was a tone, but there was no actual tone, and I am very, very confident. So I wait and I wait and I wait, and I'm eventually not rewarded. The next time, I should actually be less confident.
Mm-hmm.
So these mice are, they're not static. They should be learning on every trial. And one question I had is kind of, how do you think about this performance over a long period of time? Shouldn't if you train them for weeks longer, the number of false events should actually go down even more, right? So how do you pick that moment in this trial where it's actually relevant to what we're thinking about in terms of face validity, right?
I think that's a very good question. I was going to bring that up later on when we get to the model, more of the modeling. But whether the mouse end up stabilizing, whether the mouse ends up stabilizing, the authors actually show that they do end up kind of, with very long training, they just reach kind of steady state, suggesting that, whether when the mouse gets it wrong, when the mouse gets it wrong and reports a false positive, it's kind of maybe balanced out by when it gets it right, and vice versa. And when it gets it wrong on the other side, it's balanced by when it gets it right. And there's some kind of, the task is just difficult enough for the mouse to somehow, it's difficult enough for the mouse to maintain a steady state of kind of false alarm. It's just intrinsically that tough to tell things apart. But whether learning is involved, we will get to that in a later figure.
Can I ask you another?
Sure.
Do you think the frequency of hallucination would differ, say, among the different inbred lab strains and versus outbred wild mice?
That's an empirical question that
it might,
requires
right?
study.
It might.
Certain, right? Yeah. In fact, Ori probably have more insight than I do, but in fact, if you if you try to model what like some of the genome-wide association study has found, might correlate with schizophrenia in humans, and also what this paper has found might correlate with hallucination. You can go around looking for genetic mutations in mice that might predict we would have schizophrenia. No, not schizophrenia, hallucination. So more reporting. Sorry, Ori.
No. And I don't have any more informed answer than that, except to say that we know that other dopamine-dependent behaviors are highly variable amongst different kind of lines of inbred mice and amongst outbred mice. And there is quite a nice paper from labs that I will send around. I have to find. Now I can't remember the name, where they show very clearly that, for example, C57s behave very differently than BALB/C mice or other mice in tasks, for example, of like oh man, I've really been out of the lab. Pressing the self-reward, like pressing the lever to get a shock.
You get a shock?
To get a shock in the VTA. To get
intracranial self-stimulation.
Yeah, self-stimulation. Yes. Oh God. It's been a long time.
It has been a while, hasn't it?
Yeah.
And also, as just to nerd out a little bit more, like C57 mice, the ones that are most widely used in research, they also have early hearing loss as well, and that can be a factor. That has got nothing to do with hallucination, but just, can you even hear the sound at all? Anyway, going back to the paper, the authors have established kind of this task and show that it has some validity. Like, it appeals to our intuition that the mouse will, on some of the trials, report they heard something with high confidence, even though there was no tone, because we programmed the computer. So the authors then moved, armed with this task. The authors now further validated this task. And they asked a question: we know a couple of manipulations that have previously been found to increase hallucination. Can we use this task to catch it? So actually, Vincent earlier asked, does this task induce hallucination by itself? We don't know, but there's a manipulation that has been reported to increase hallucination in human. And that is if you just, if you keep pairing the appearance of a picture with a certain tone to human, and you keep pairing it over and over again, then what people have found is that you can remove, I can't remember either the tone or the picture, the person can still perceive the other thing that's missing. They can kind of fill in the blank. And this effect is increased in schizophrenia patients that are undergoing hallucination. So this kind of like binding of stimuli is kind of similar to, I don't know whether you guys heard of this experiment where they have human subjects looking at a rubber hand and then putting their own hand under the table, and the experimenter would have a paintbrush and they would brush the rubber hand, and at the same time someone else would brush the subject's real hand under the table, and do it a few times, and then the experimenter would get a hammer and smash the rubber hand, and the subject would be completely freaked out because they have now associated the paintbrush sensation of their own hand to the rubber hand. So they thought the rubber hand is theirs, and they completely, they flinch, and, they get very scared when you break what they thought is their hand. So this kind of binding, people have shown, in research, in some other research setting, to be potentially increase in schizophrenic patients. And it's learned because you have to undergo the training of the rubber hand and the paintbrush, or like the visual stimulus and the tone several times before this effect is seen. So based on this human finding, the experimenters proposed that in their mouse task, if they increased the number of trials where there is a tone, so they increase the proportion of trials that there's a tone, that might increase the mouse's propensity to think there's a tone when there's no tone and be confident about it. And that's exactly what they found, that the percentage of false positive that the mouse makes with high confidence was increased scales to how often the mouse actually hears a tone. And this is important in that the mouse, when they make these mistakes, they are actually losing out on water. These are actual mistakes. So the fact that the mice are still doing it suggests that maybe, well, it's not reward related, but maybe something is really changing in the perception of the stimulus.
Although, okay, you could also argue that in those sessions, they get more water rewards because there are more correct choices, because there are more sounds, right? So therefore, they're less motivated to get the water, and they therefore are reporting their confidence differently than in other sessions.
That's very good.
That makes sense.
That does make sense. That's
I guess but on the other hand, they're being rewarded for the correct choice, whether there is more water, whether there is a true tone or not. So, I retract my statement. It should be fine, right? Because if they're getting the reward for a correct choice, regardless of whether there's a tone or not, whether they're correct because there's a tone or whether they're correct because there's an absence of a tone, doesn't change the number of rewards that they actually get.
That's true. And there's another measure that these authors did that was very, that's a nice control, which is if you imagine the mouse is getting more water reward and therefore is no longer thirsty and they just had enough water, you might expect that they, not only would they respond less, they might also be less willing to wait around in the water port afterwards. So they might, the entire distribution of how long they're waiting, the time investment might go down, or the whole thing might shift. But what these authors did was they did a median split of how willing they are, how long they wait at the port for the reward, and anything above the median they classify as high confidence trials. And of these high confidence trial, they still see more false alarm. So if the time investment as a whole is changed, the whole distribution is changed, then this median split would not show an effect, but they still find an effect. I didn't explain that very well, but sorry about that. Go read the listeners, please read the paper. That was a poor job. But anyway, there's also the authors also track how far. I think they track other measures of whether the mouse is motivated for the task or not, and it seems fairly steady throughout the task. So they think it's okay. But anyway, so that was manipulation number one to see whether you can kind of increase these hallucination-like choices. They call it hallucination-like percepts, which is just high confidence false alarm. So the second way that the authors did to increase these hallucination-like percepts that they know works for human is by giving a drug called ketamine. And ketamine is interesting because it is now it is known in large doses. Well, first of all, in very, very large dose, it's used as in the UK, known widely as the horse tranquilizer, is used as an anesthetic in animals. So like in mice, when we anesthetize mice, we sometimes use ketamine as well, and horses, and also in humans. But in slightly lower doses, it can induce hallucination. So in sub-anesthetic doses, it can induce hallucination, and it works by blocking this glutamate receptor called NMDA receptor, and it's related to an old drug that was abused in the 70s called angel dust. I don't know if you guys heard about this. So angel dust it's now completely fallen out of fashion, but back in the 70s, it was widely abused, also known as PCP. And that drug also blocks NMDA receptors and it triggers, hallucination. And so does ketamine. And interestingly, ketamine at even lower doses is also used as an antidepressant. So drugs have very dose-dependent, differing effects, which is fascinating. We don't know why. But these researchers found at a dose that doesn't trigger anesthesia, can increase these hallucination-like percepts. So these are two kind of these are two just positive controls to tell you that this task is probably testing for hallucination. It has, now, as already said, has certain predictive validity because in humans, manipulations that causes hallucination, they can recapitulate it in mice using this task.
I would say that this has construct validity from this experiment, right? So now we're taking something that we know causes hallucinations in humans and we're putting that into this mouse model. So therefore, the same constructs, so the same neurobiological constructs are mediating this behavior in mouse as are mediating the behavior in humans.
Got it. So predictive validity is if we were to find something in mice that change that we can replicate in human, then it's predictive validity.
Yeah. So if
it's the other way around.
Yeah, exactly.
Okay.
I mean, it's kind of hard to separate. I would say that one thing that was very nice about this experiment was that they show, so essentially ketamine has a quite a short half-life. Mm-hmm. And a very nice internal control was that they show that they only see an effect on the number of hallucination-like percepts in the very beginning of the experiment. So this is figure 2M. So by the end of the session after they've been treated with ketamine, they actually look the same as if they were treated with vehicle because the ketamine has washed out. And this is a very, very strong analysis that shows that the change in false alarms or hallucination-like percepts is actually caused by ketamine itself and not something else.
Yep. That's a nice analysis.
The only thing I would wonder, and then I know we're getting really into the weeds and we can potentially speed up, is that, if you look at the number of trials that mice are doing with and without ketamine, it's very different. So the overall structure of the session is probably quite different. Mm-hmm. It's not as if they're just saying, oh, I have a higher confidence in the false alarms, but that they're actually either kind of initiating fewer trials, they're preoccupied internally in some other way during the session. And that I wasn't able to kind of see kind of a deeper analysis except in N and O, where you see the number of trials that mouse K18 does is about half as in O as it is in N when it's had ketamine. And
I didn't even notice that.
A similar thing is, so in E through G, the level, the percentage of high volume sounds changes, right? And you can see the number of trials is actually interestingly changed depending on how many high confidence essentially events that they have. So there are many different things, but I think that 2M is really a strong support of their idea that these are kind of ketamine-induced perceptions.
Right. Yeah. And you can imagine that ketamine by itself, like high dose of ketamine, can also affect the ability to perform the task and also like some sort of motivation. Yeah.
Exactly.
Yeah.
Exactly.
Isn't ketamine used to treat certain psychiatric disorders, Ori?
So low dose ketamine is used as a rapid acting antidepressant, and that's been recently. And in fact, that is actually limited by the experience of hallucinations during the infusion. So, for example, sometimes when you get this infusion, you have a relatively immediately high dose that then kind of washes out as it normalizes, and that can actually have distressing hallucinations. So sometimes that can be limiting in the dose that's used.
So it blocks subsequent antidepressant effect if it triggers hallucination.
No, in fact, well, this is a complicated literature that I definitely don't know a lot about, but actually some people believe that dissociation during the initial infusion is actually correlated with the long-acting antidepressant effect. So this is the kind of kinetics of ketamine as an antidepressant are very interesting. So if you think about an SSRI, and I'm sure some of our listeners have gone to the doctor and felt depressed and got an SSRI, and the doctor will say, okay, take it every day for 4 to 6 weeks and you'll come back and maybe you'll see an effect then. But ketamine is actually, you see an effect with a single infusion that can last weeks without taking it every day, essentially. And so it acts through a different mechanism through some sort of plasticity in the brain. And it's very useful in kind of acute psychiatric emergencies where you may not have time to wait for SSRIs to take effect.
Cool. All right. So, going back to the paper, we might have to speed up a little bit. Yeah. So going back to the paper, the authors have found that this mouse model of reporting whether there's a tone or no tone with a confidence, time investment confidence, seem to recapitulate a lot of humans, two of human data. They now went back to humans using a similar design and see whether a similar design in human can actually correlate with hallucination phenotype. So I'm guessing this particular model has not been applied in humans yet as a diagnostic. So what they did was they designed a task that can be completed online, which mirrors what the mice did. So the task involves a human putting on a pair of headphones, and they have to detect whether a tone has been played, and the tone is, again, embedded in white noise. And then the human would have to, click with the mouse. Did you hear a tone or not? Yes or no? And after this decision, the subject would have to decide on how confident they are on a slide bar. So instead of time investment, the human just have to self-report how confident they are. And after that, the human gets a feedback in terms of points, in whether they have been correct or not. So the authors basically crowdsourced this online task via this thing called Amazon Mechanical Turk, which is basically a big crowdsourcing service that you can do. And they got a lot of, I can't remember.
220
participants. Okay, thanks. 220 participants and many, like tens of thousands of trials. And they got results from humans about how they performed. And they found that with humans, even on trials that doesn't have any tone, they reported there was in fact a tone 7% of the time. So even just like in mice, humans have sometimes would report a sound when they didn't hear a sound. And in this experiment, they didn't just in addition to this online task, the researchers gave the participant after they finished the task a questionnaire that asked them a bunch of questions on how often they have hallucination. So it's basically a previously validated questionnaire done by a schizophrenia researcher that has a panel of questions that ask about, do you perceive, smell or sound or sight sometimes stronger than you would expect? Do you hear sound when there's no, obvious explanation of that sound? Do you hear sentences, voice, that kind of stuff? So there's a panel of all these questions. And the researchers basically found that, and these panel of questions can be kind of separated into different kinds of score, for example, hallucination score, but also things like depression score and anxiety score. And what the researchers found was that the hallucination score was the best predictor of how often these subjects reported false alarm with high confidence. So this suggests that this online test that they did, that they kind of mapped from the mouse model, actually does predict, I guess. Is this correct? Yeah, I'm going to say yes. Does predict hallucination phenotype in human. At least it correlates with it. And what's interesting is that, as already mentioned earlier, these are just crowdsourced humans, and most of them, if not all of them, I actually can't remember, but vast proportion of them do not actually have schizophrenia and do not have diagnosis of suffering from auditory hallucination. But yet you still see a spectrum of both on the task, this high confidence false alarm rate, and also on the hallucination questionnaire, like a big spectrum of how often they have these hallucination-like percepts. So it points to there existing a spectrum of these kind of hallucination-like audio sounds, not just for cats, but for arbitrary tone played on the internet.
And I think that this now gives us some excitement about thinking about if we can understand the underlying biological mechanisms that control the mouse's confidence in whether a sensory experience is true or not, then we are understanding maybe the same mechanisms that are controlling whether a human is having that sensory experience or not. I think that the question that I have is that are sensory experiences that are not real, that we're highly confident in, would the mechanism of that be the same in someone who's having hallucinations and that are causing impairment in a psychotic disorder?
Yes.
Or is this just a trick of the brain that is present in everyone? And, the mechanisms that are present in a quote unquote normal brain are not relevant at all to psychiatric disease. And this paper did not convince me that is the case here.
Yeah, I had the same thought, but I wasn't confident enough to ask it because I don't know anything. But I agree that maybe these are just not relevant to pathologic hallucination, right? Yeah, it's a good question.
And I mean, the problem is that in mouse, what are you going to use to test that, right?
Yeah.
There's no mouse that has kids, but they have human data. So it would be so nice if I've done this in a population of patients with schizophrenia, but of course this is a massive undertaking, and I'm sure something that Katarina Schmack, who according to Twitter is a psychiatrist and neuroscientist, is.
Sorry, or you cut out. Could you repeat the last sentence?
Oh, sorry. And I'm sure that this is something that Katarina Schmack, who's the first author and is a psychiatrist in neuroscience, is working on as we speak, is applying this task to patients with schizophrenia who are psychotic or even patients who are at high risk for schizophrenia. So this has been a huge push of NIH is to essentially take the first psychotic episode and try to intervene on them before they develop full-blown schizophrenia in their teens and 20s, and whether you could then risk stratify them further by having them from this task.
Yep.
Do you think there's no better human task for predicting hallucination? I don't know whether you know, because you're not a psychiatrist, you're a neurologist, but
I'm definitely not a psychiatrist. I guess my question, and I haven't quite settled on what the answer is yet, but is this any better than pre-bolus inhibition in terms of developing new drugs and thinking about kind of what the next phase of schizophrenia therapies are for the positive symptoms, which are psychosis and delusions, right? And if you have to train your mouse for weeks and weeks on this complicated behavioral task, is that really the answer? Or are we finding something fundamentally new compared to these tasks that we can do, that I could do, who's someone who's a molecular biologist and an electrophysiologist, that I can throw a mouse in the box and do pre-pulse inhibition. Right?
I think, I think that is going to be future work for this paper, for sure. Like, this paper is very much establishing the baseline paradigm, I think.
Absolutely.
Yeah. But having said that, let's go to the next experiment that these authors did that further establish some sort of validity. I think this would be, again, construct validity, which is they've shown that this kind of task maps to human hallucination as well, not just maps to human hallucination. So it suggests if you see something in mice, it might tell you a little bit about hallucination. Now, one major neurotransmitter that's been implicated in schizophrenia is dopamine. And the reason why there are three, as far as I can tell, I might be completely wrong, Ori, please correct me. There are three classes of neurotransmitter that may be implicated in schizophrenia. The first one we talked about, which is NMDA receptors and therefore glutamate. So if you block NMDA receptors, you can get hallucinations. So suggesting that glutamate can be involved. The second one is dopamine. And the reason why people think that is because, first of all antipsychotics that are prescribed now have the ability to block dopamine receptors, particularly D2 receptors. So it suggests that dopamine is somehow involved because the drug that treats it blocks it. And also there are some human PET scan studies, positive positron emission tomography, that uses radio tracer that binds to dopamine receptors that found dopamine release can be affected in schizophrenia, and also dopamine receptor can also be changed by schizophrenia. So it strongly implicates dopamine. The third one is actually, I think, less well studied. It's serotonin. And the main reason is things like LSD causes dramatic visual hallucination. But what's interesting is that some of the more recent antipsychotics, the atypical antipsychotics, also have serotonin receptor activity as well. So it might work on both. But these authors then decided, having armed themselves with this new task of being able to tell whether mice are reporting false alarm with high confidence, so hallucination, they think, they decide to look at the brain. What is different in the brain when the mouse are reporting that there's a tone when there was no tone? And the way the authors did that was to image dopamine, to look at dopamine levels in the mouse. So as I said just now, in humans, we look at dopamine levels by PET scan. It's a little bit more difficult to do that in the mouse because for PET scan to work, you have to be completely still. Otherwise, the image would smear and smudge. But in this task, the mouse is moving around, sticking its head in this port and that port. So the authors use this relatively new technology called dopamine imaging. And how this works is this. Researchers have recently designed, so listeners might have heard of this fluorescent protein called green fluorescent protein. And this protein would glow green when you shine a blue light on it. But you can actually design this GFP, green fluorescent protein, to report certain signaling event has occurred. So what these researchers didn't do it, but previous researchers did, was if you chop a GFP protein right in the middle in half, that actually completely stops the fluorescence, stops the GFP's ability to fluoresce. But if you then take the two ends that you chopped up and you kind of link it to a dopamine receptor, what then happens is when the dopamine, where in this new kind of chimeric dopamine receptor that acts as an optical sensor, when dopamine binds to this GFP-linked dopamine receptor, the dopamine receptor undergo a conformational change, and it kind of brings the GFP molecule back together, and now it fluoresces. So using that, you can use how much the fluorescence level, how strong the fluorescence level is, as a measure of how much dopamine there is. So
And this is perhaps just to go back to kind of our arc of how new technologies have changed neuroscience. This is something that so in 2018 there was I think Tim and I were both at this basal ganglia Gordon conference when this was first. It might have been 2016. I think it was 2018 when this was first shown in a talk. So these are called GrabDA. And these are kind of, we were all, I remember sitting there and people were just shocked and saying, wow, this is going to revolutionize the way we study dopamine levels in the brain in animal models.
Yeah, its earlier cousin, which is GCAM, calcium, for detecting calcium. That's completely revolutionized how people look at the brain. Yeah. So this allows the authors to basically express this GrabDA, this dopamine sensor, inside the striatum, and then stick in a optic fiber so that you can pulse in blue light and then detect how much green light gets fluoresced back up. And the idea is the more dopamine is released, the more green light you can detect. And the authors tracked dopamine levels throughout the entire trial and then divided it into if the mouse was responding correctly, incorrectly. Was it a false alarm? Was it a false alarm with very long time investment and therefore high confidence? So just to speed things up a little bit, I'm going to be summarizing quite a lot. There's a lot of work gone into this next figure. The authors found was that when you present a tone, as expected, the tone itself can generate dopamine release. And this has been previously been reported. And the louder the tone, the stronger the dopamine release. But what was interesting and what the thing that, the thing that the authors were interested in was not just when the tone was being played, but the state of the animal as it was going into the center port. And the reason is this. I didn't quite explain it during the last when we're discussing, I didn't quite explain it. But one of the model for why the mouse is reporting more false positive with high confidence is that it is, like Ori said with his cat, the mouse might be expecting to hear a tone more often than an ideal observer would. So one way of thinking about this, I'm gonna spend a tiny bit of time talking about this thing called signal detection theory. Actually, you know what? I might skip this because how much time do I have left?
Whatever you want. It's up to you guys.
So I will. Okay,
let's do it.
Okay, let's do it. So
if people haven't left already, they're not going to leave now.
Okay. So this whole paradigm of detecting false alarm and whether you detect things correctly or not was developed to analyze radar back in Second World War. So as you can imagine, back in Second World War, when radar was first developed, it was for detecting whether there are air raids coming to London to bomb, well, not just London, bomb UK. And to be able to detect radar, it is actually slightly difficult because the radar operator needs to be able to tell whether it is a plane coming or whether it is just a flock of birds coming across the channel. And those can look kind of similar. And without training, you need a little bit of training to be able to tell them apart. And with some training, without any training, you'll be, making a lot of mistake. You have a lot of false positive, but you also, so when it's actually, a flock of birds coming, you might mistake it as a plane. And also when a plane is actually coming, you might think it is a flock of birds just because you haven't been trained. With training, you can get better at discriminating the two signal apart. But that is not the only thing that can affect your judgment. Another thing that can affect it is your expectation. So, for example, if someone comes in with intelligence saying that, we just have some intelligence that a bunch of planes have left the airport and is crossing the channel, you might now be on high alert, and you might be much more likely to call any blobs as a plane because you're expecting that planes are coming. So in that case, you would have more both correct report of correct planes, so people called hits, but you also might have increased false alarms. So you might be much more likely to see a blob as a bird, to see a blob as a plane, even though it's a bird. So one analogy is that, for example, if you are in a haunted house, if you go to a haunted house, or if you go to a cabin in the wood by yourself, you might actually start hearing weird noises, even though there's nothing there. So your own internal sense of belief can actually drive some of your decision and some of your perception of outside noise. So what these authors are testing here is whether the dopamine level in your brain in the period right before you even go in the center port, whether that would predict whether you have hallucination-like percept. And they found, in fact, that that's what they found, is that if you have higher baseline dopamine as you go in the port, then you're much more likely to have high confidence false alarm. So higher probability of hallucination, which makes sense from what we have found in humans, that schizophrenics have higher dopamine release. What is
and it makes sense in the sense that dopamine antagonists that antipsychotics are dopamine antagonists. So you can imagine that it lowers the level of dopamine immediately before the stimulus or the tone, and therefore lowers your kind of expectation or your brain's expectation that the tone is actually going to be there.
Right. Exactly. And what is quite interesting is that they found that this prediction that your prior belief that there would be a tone, the prediction that the increased dopamine correlates with your prior belief is especially found in this newly rediscovered region of striatum called the tail of striatum, where researchers in the last 5 years have found to be an area especially important for detecting the salience of external stimulus and also like how threatening they are. So all this fits very nicely with that literature. So it's really nice. What is somewhat interesting, though, is that the authors didn't quite report when you do hear a tone, what is going on with the dopamine level. Like on the trials where you actually are, on the trials where you're about to make a false alarm choice with high confidence, do you actually perceive the tone as higher in terms of your dopamine level? So that's something that might be interesting to look at. And as Ori and also Vincent asked earlier, just because this task can tease out these high confidence hallucination-like percepts, is the mouse actually hearing something inside his head? It's still not quite yet established. So, for example, in humans, they have found that when human subjects are hallucinating, undergoing auditory hallucination, there are some part of the auditory cortex in fMRI, it actually sometimes lights up. So when a mouse is undergoing this task and it's hearing a false alarm with high confidence, is it also not just the dopamine system, but like is other bits of the brain also lighting up? It's not yet known. But this task allows the researcher in the future to like look at both regions simultaneously.
Tim, should they have used a dopamine antagonist and see if it affected their measurements in mice, you think?
They should, and they did in the next figure. So in the final experiment that these experimenters did was the previous experiment we discussed was they looked at how dopamine level changes as the mouse is making these hallucination-like choices, hallucination-guided choices. So it's correlative, but to really tie the story up, you want to be able to perturb the system to change things yourself and see whether the behavior also changes in the way that you predict. So in the very last experiments, these researchers used optogenetics to actually activate these dopamine neurons in the tail of striatum in the region where they found increases your false percepts. And they showed that these mice, when you activate dopamine like this, these mice make more of these false alarm with very high confidence. And this effect is blocked by antipsychotic, which is a dopamine D2 receptor blocker. So based on all this, they think that their task, they concluded that their task likely is telling you whether the mouse is hallucinating or not.
So I think one interesting experiment, and these are obviously incredibly difficult, would be to have looked in the mouse that had received ketamine at these dopamine levels, right? So that would have tied together kind of a non-dopaminergic initiator of hallucinations with this dopaminergic mechanism that they're postulating is responsible for the hallucinations in the tail of the striatum. And that would have given perhaps more construct validity to the last part of the paper where they get at the neurobiological mechanism, that this neurobiological mechanism is actually what's mediating hallucinations when the hallucinations are caused by a trigger that we know causes hallucinations in humans, like ketamine.
Do people know whether ketamine-induced hallucination or PCP-induced hallucination is treatable with dopaminergic antipsychotics?
I think that they are, but I will go back and check. And I think that kind of to just take a big picture look at this. So we can, this has been a thread throughout our conversation about how this compares to pre-pulse inhibition or other models of psychosis or hallucinations. And on the one hand, the question is, what do we learn from this? And this is kind of what I'm thinking of, or what is the added benefit of this paper to this kind of to developing new treatments for schizophrenia? And I think the answer to that is that we don't know yet. And I think that hopefully in the future, they'll start to test this about whether this task is predictive of future conversion to kind of impairment, whether this task is related, like is changed in populations of patients where we know that they're already schizophrenics. And also whether we could start to do some sort of kind of translation of the mechanism to drug targets or something like that. You could imagine that these mechanisms are all very interesting. But if you could actually target one part, dopamine release in one part of the striatum, but not another, you could actually affect hallucinations, but not motivation, for example, and have a really, really elegant kind of way to treat some of these impairing and kind of the symptoms that are impairing without affecting kind of everything, which is what we know antipsychotics do, which is that they can decrease motivation and kind of affect behavior outside of hallucinations as well.
Yeah. Agreed. Yeah. One thing that it would be nice if the author did, they didn't quite do this, was that, as Ori alluded to, there are different regions of the striatum that might mediate different kinds of behavior. And they didn't. It would be nice to see whether if they were to optogenetically stimulate other bits of striatum, would they, does this hallucination-like behavior does it only reside in the tail striatum and not in other parts of the striatum? That would be nice.
And the limit of this behavioral task is that it is so difficult to do and to train the mice that you are actually limited in the number of experimental manipulations you can actually do, right?
Yeah.
I imagine that the reason that they didn't do this, or they didn't use amphetamine as well as ketamine to induce hallucinations in these mice, is that it is just so time consuming to do. And you can actually see in one part of the paper, they say, we focused our analyses on the tail of the striatum instead of both regions of the striatum for efficiency, essentially. So this kind of, when you think about an animal model, actually is a detraction from this animal model as opposed to pre-pulse inhibition, where you can do it in one day, right?
That's true. Although, if the thing that you are trying to model and trying to test is itself complicated, it might just be the price to pay. Like, it's unavoidable that you'd need a lot of work to be able to model that. In which case, it would just suck.
Yeah. I agree. I'm not convinced yet, because they haven't done the next experiment to show me that that's actually true, right? If they could show me that something that they found in this model that isn't there in the pre-pulse inhibition, I would be very excited. But this is, of course, probably going to be the life's work of Adam Kepesh and Katarina Schmack, right? Yeah. And that's exciting.
Anyway, so that's that. I think pretty much wraps up this paper. I don't know if you guys have any more questions.
This was all a hallucination.
Yes.
Someone asked me once, how do you know this is not a matrix style construct, which I guess is an induced hallucination, right?
It's right, exactly. It's I don't think it's possible to tell. Or whether
unless there's a glitch, right?
You have to find glitches. Yeah.
Well, that's the thing about knowing if you're hallucinating.
That comes around.
The cat. If you look for cues for realities, right? Reality checks, and it's what you said at the very beginning. How does Ori know that this is real? You look for cues that reinforce it, right? I think that's a part of it.
Right, but what are these cues? It just has to be consistency. That what happened yesterday is happening again today, But if you're so one interesting thing, I'm going to finish this. I'm going to finish this with a story. Finish this paper with a story, which I've completely lost my train of thought of. I can't remember how it relates to it. We were talking about consistency. But what if you from birth you have hallucination and it's just consistently inconsistent, then it will be very, very difficult to. But the story I was going to tell, which relates to, I'm just going to tell the story first and then talk about it. So one of the reasons why this paper caught my eye was I remember when I was an undergrad in the UK, I went to a lecture by a guy called Simon Baron-Cohen. And Simon Baron-Cohen is famous for two reasons. One is that he is a very famous autism researcher, actually, in the UK. And the second reason is that he's a cousin of this guy called Sacha Baron-Cohen, who in the UK is better known as Ali G, and in the US is better known as Borat. So in this lecture from Simon Baron-Cohen, he was talking about schizophrenia. And he described the following scene. Imagine you went hiking with a friend, could be your brother, and you were separated for like 2 hours. And then eventually you found your friend, and he's in front of a bush on his knees, mumbling to himself. And then he turned to you and he said, I've just been spoken to by this tree. The tree was on fire, and it told me he told me that it is the voice of God, and there are these lists of things that I need to do. Listen to me, follow me, and listen to what I say. Follow all these things I have to do. So Simon Baron-Cohen said, "Well, do you think this guy is hallucinating and seeing things that aren't really there, or do you believe him and kind of follow him to the desert for the next 40 years?" Because he's alluding to, of course, the story of Moses, who was, talking to a burning bush, and then, Exodus happened. So that really kind of struck me because, first of all, Moses was like the most metal of all character in the Bible, with the plagues and river into blood and stuff. But also, it shows it ties to why schizophrenia is even in the population. Like, why do we see schizophrenia? So unlike a lot of other neurological diseases, such as, Parkinson's and Alzheimer's and even Huntington's, all these neurological diseases are devastating, but they kind of appear after reproductive age. They appear as you get old. But schizophrenia is different. It is, by all account, devastating. It's very difficult to have a normal life if you have schizophrenia and function normally. But it actually appears around late teenage and early adulthood, like at reproductive age. So if it is so devastating, why is it still in the population, and why has natural selection not why is it not a burden on natural selection? And Simon Baron-Cohen's answer was that it is actually schizophrenia. Is actually, like Ori said earlier, and as the questionnaire showed, is actually possibly a spectrum. So it exists. Some people might have more propensity to hallucinate and hear things, and some people might have less. And there might be a Goldilocks zone of kind of hearing things that aren't there that might provide an evolutionary advantage, because you might be more convinced about what you hear and what you see, and therefore act upon it more. Like, finally, I'm gonna go to the gym and stick to go to the gym because I have this, perception of exercising is good. I'm gonna stick to the diet. I'm gonna go to the desert and find the promised land. That kind of conviction might have evolutionary advantage, but too much of it might then be disadvantaged. And from, just a random chance of both genetics and environment, you would get, the extreme end of this, and you get the schizophrenia. So, I just thought I'd tell that as a cool story involving Borat's cousin.
Very good. That was great. Thank you, Tim. I really enjoyed that. This idea of trying to figure out what the mice are doing is really compelling, I think, right? All right. That is TWiN 21. Show notes are at microbe.tv/TWiN. And there we're going to have a link to a poll which we'd like you to go fill out if you haven't done so already. Give us some feedback on what we're doing here and what we should be doing and so forth. And as Rory said, maybe you get a prize at the end. So that'll be at microbe.tv/TWiN. We have a backlog of email we'll get to at some point. But if you have comments, questions, twin@microbe.tv. And if you really like what we do in this whole family of science podcasts called Microbe TV, consider supporting us, microbe.tv/contribute. Ori Lieberman is at the University of California in San Francisco. Ori Lieberman on Twitter. Thanks, Ori.
Thanks, Vincent. Thanks, Tim. This was phenomenal and excited to be back.
Good. Tim Chung is at New York University. Thank you, Tim. Great job.
Thanks, Vincent. Thanks, Ori.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll see you next time.